Hello students. Now in this session we are moving on with the same chapter, that is sexual reproduction in flowering plants. Now in the last session we had finished with the androecium, that is the male reproductive structure. Now in this session we are moving on to study about gynoecium. Gynoecium is the female reproductive structure in a flower. Okay, so gynoecium in a flower, I told you in the last session that gynoecium consists of units called as pistils. Okay, so pistils, or you can also call it as carpel. So we'll start with the study. So this female reproductive structure, okay, now we are going to study about pistil, okay, and megasporogenesis, and also about the embryo sac. Okay, so these are the things we are going to study today, that is under gynoecium. So now when I told the female reproductive structure, I told you it consists of units called as pistil. Now in a flower there may be only one pistil, or some flowers we see that it has more than one. There may be many pistils. Okay, so when in a flower, when there is only one pistil, okay, we call it as monocarpellary. Okay, so that is the term we use to describe a condition where a flower has a single pistil. Okay, so then we call it as monocarpellary. Okay, so that is a term. Now in some flowers we find that the pistil number may be more. There may be more than one pistil. When there are more than one pistil, then we call it as multicarpellary. Okay, so monocarpellary and multicarpellary are the terms we use to describe about the condition of gynoecium, whether there is only one pistil or whether there are more than one pistil. Okay, now when there are more than one pistil, okay, then we used to describe it in different way. Now when it is multicarpellary, so you know the meaning of that one. Multicarpellary means more than one pistil. So when there are more than one pistil, the carpels may be fused. Okay, many pistils, it may be fused, and then we call it as syncarpous. So when we say syncarpous condition, it means that all the pistil or carpel, they are fused together. There is another condition where we say apocarpous. So when there are many pistils, all the pistils may be free. Okay, it may not be fused to each other. When they are free, we call it as apocarpous. Okay, so that these are the two conditions which we find in a flower if it has more than one pistil. That is, it may be syncarpous or it may be apocarpous. So that you need to remember these terms, that is monocarpellary, single carpel, multicarpellary, more than one carpel. In a multicarpellary condition, if all the pistils or carpels, if they are fused, then we call it as syncarpous, and if they are free, then we call it as apocarpous. Okay, now that is about these terms. Now we'll move on with the study of pistil. Okay, so pistil or carpel, so we can use any one of the term. So this is a structure of a single pistil. Now you can see it has a structure called as stigma. So stigma is the part which we say that it's like a platform for the landing of pollen grains. Okay, that is called stigma. Beneath this you find that elongated structure called as style, and the basal bulged part which we call as ovary. Okay, so pistil we divide it into three regions, that is stigma, style and ovary. Okay, now within this ovary, inside this ovary, we find there is a cavity. Okay, there is a cavity which we call as locule. Okay, so locule is the cavity which you find inside the ovary. So there's like a space, a cavity which you find inside the ovary, which we call as locule. Okay, now within this locule you will find there is a tissue inside which we call as placenta. Okay, now placenta is a tissue which we find inside the ovarian cavity, that is inside the locule there is a tissue called as placenta. Now in your last year's syllabus, if you just recall, you had studied about placenta, and I hope you remember the term placentation. Okay, so placentation is a mode of arrangement of ovule within the ovary. Now what is this placenta? It's a tissue to which you will find that ovules will be attached to. Okay, so placenta is a tissue which you find inside the ovary, and from this placenta, from this tissue arises ovule, or we also call it as megasporangium. Okay, so commonly we use the term ovule, okay, or we also call it as megasporangium. Now this ovule, you will find it inside the ovary attached to the tissue called as placenta. Now about this ovule, you should know that ovule, it may be only one in number or it may be many. Okay, it depends from species to species. Now only one ovule inside the ovary, like for example wheat, for example rice, okay, all these have a single ovule inside the ovary. Okay, or some other examples like papaya, watermelon, all these will have more than one ovule inside the ovary. Okay, so it may be the ovule may be one or many depending from species to species. Okay, so that is about the structure of a pistil. So we studied about the structure of a pistil. Okay, now I told you inside the ovary there is a structure called as ovule, or we also call it as megasporangium. So now we are going to study about the structure of megasporangium or the anatropous ovule. Now ovule, it is of different types. Okay, we are going to study about a typical anatropous ovule. Now I told you this ovule, it will be inside the ovary attached to placenta. Okay, now this structure we are going to study, and this is a 5 marks question. Okay, so 5 marks question where you will be asked to explain the structure of a typical anatropous ovule, or this term can also be asked as explain the structure of megasporangia. Okay, so now this is the structure of a typical anatropous ovule or the megasporangia. So you see that it has a stalk. Okay, it has a stalk which is called as funicle. So this stalk will be attached to the placenta inside the ovary. Okay, so now this is a stalk which is called as funicle. Now funicle, that is the stalk, is attached to the body of the ovule. Okay, so this is the body of the ovule and this is a stalk. Now this stalk and the body of the ovule, you see a junction. Okay, a junction where the stalk is attached to the body, and the junction we call it as hilum. Okay, so hilum is the junction between the stalk of the ovule and the body of the ovule. Okay, now coming to this part, the body of the ovule is covered by 2 layers. Okay, 2 protective layers which we call as integuments. Okay, so integuments, you can see I have labeled it here. 2 layers are there. Outer layer is called outer integument and the inner layer is called inner integument. Okay, so outer integument and inner integument. So it is two layers covering the body of the ovule. Now if you observe these integuments, it is not covering the whole body. Okay, so you can see that in one region, okay, it is not covering. So that part we call it as micropyle. So micropyle is the opening or the place where the integuments are not meeting there. Okay, so there is an opening there which we call as micropyle. Okay, so this opening we call it as micropyle. So here you can see the integuments are not fully joining. Okay, so there is a gap there, there is an opening which we call as micropyle. Okay, now within this integument, so you can see outer integument, you can see inner integument. Inner to this, you will see a mass of tissue which we call as nucellus. Okay, so nucellus is this mass of tissue. So you can see here, this mass of tissue we call it as nucellus. This, so where I am shading, so this region we call it as nucellus. So nucellus is a mass of tissue which you find within the inner integument. Okay, now the cells of nucellus, within the cells of nucellus you will see a structure which is embryo sac. Okay, so embryo sac can also be called as female gametophyte. Okay, now this embryo sac inside, so this structure here we are not showing the details of embryo sac, that we will study later. Okay, so here we are just showing the overall structure. Embryo sac will contain, okay, the egg cell. Okay, so that in detail the study we will do next. So now you see within the nucellus you will see this structure called as embryo sac. Now this embryo sac is present towards the micropyle. Okay, therefore we call this region as micropylar region. So micropylar region means the region which is like towards the micropyle. So embryo sac is present towards the micropylar region. Now the opposite end of this, now this is micropylar end, the opposite end, this portion we call it as chalazal pole, or we call it as chalaza. Okay, so that is the opposite end, that is opposite of the micropylar end. Okay, so this will be the structure of a typical anatropous ovule. Now as I told, when a question is asked for 5 marks, you need to draw this diagram and also explain its parts. Okay, so the funicle, which is a stalk, junction is called as hilum, and the body of the ovule covered by 2 integuments, outer integument, inner integument, and within this, and it is it has an opening at one end called as micropyle. Within the integuments you find a mass of tissue called as nucellus, and within the nucellus you find the presence of embryo sac. And I told you embryo sac is also called as female gametophyte. Okay, so this will be the structure of a typical anatropous ovule. Now we studied about the structure of ovule. Now next what we have to study is this, that is embryo sac or the female gametophyte. How does it develop inside the ovule, and what is the structure of the embryo sac? So both we will have to study, and that we will study now. So we finished studying about the structure of a typical anatropous ovule. Okay, so now I told you within the ovule there is a structure called as embryo sac or the female gametophyte. Now what we are going to study is how did that female gametophyte develop inside the ovule. So that we are going to study now. Okay, so that is, next we are studying about Megasporogenesis. Now what is Megasporogenesis? So it's a 1-mark question. So we say megaspore mother cell, okay, undergoes meiosis and produces megaspores, or you can also say in the other way, production of megaspore from megaspore mother cell through meiosis. So this process we call it as Megasporogenesis. Now I have told some new terms here. I have told you megaspore mother cell, I have told megaspore. So what is all that we will study now? Okay, then I will come back to the definition. Now I told the word Megasporogenesis, that is megaspore mother cell. Now what is this megaspore mother cell? Now when the ovule is young, okay, when it is young, in the nucellus, one cell will become megaspore mother cell. So what am I explaining here is how did the female gametophyte develop? Okay, so that is what we are studying now. So when the ovule is young, in the nucellus, you will see that one cell becomes megaspore mother cell. So it becomes larger in size, okay, and it has a lot of reserved food materials. It will have a prominent nucleus. Okay, so this one cell is called as megaspore mother cell. So it is developing where? It is developing inside the nucellus. So in the nucellus, one cell which is almost towards the micropylar end, okay, one cell becomes megaspore mother cell. Now this megaspore mother cell is undergoing meiotic division. Okay, so it undergoes meiotic division and it will produce megaspores. So when I say meiotic division, so how many megaspores will be formed? Okay, so you know that 4 megaspores will be formed. So you know the difference between mitosis and meiosis. So last year I have taught you about the difference, mitosis and meiosis. So here meiosis happens. So one cell undergoing meiosis, you know that 4 daughter cells are formed. So that is what is happening here. One cell undergoing meiosis, okay, and it is producing 4 daughter cells. So we call those 4 daughter cells as megaspores, and this development we call it as Megasporogenesis. Now I told that meiosis happens. So why do you think only meiosis should happen here for the production of megaspore? What is the importance of meiosis here? Okay, so again you need to recall your last year's syllabus where I had taught you that gametes are always haploid, right? So gametes are always haploid. Now in this chapter we are studying about angiosperms. Okay, so you know that we studied about so many plant groups in the last year's syllabus: algae, bryophytes, pteridophytes, gymnosperms and angiosperms. And angiosperms, the plant body is sporophyte. Okay, so plant body of angiosperm is sporophyte. So when it is sporophyte, you know that sporophyte means all the cells of the plant body is they are diploid, or we say 2N. Okay, so I hope you can recall all that. So when every cell of the plant of angiosperm is diploid, including megaspore mother cell, this is also diploid. If this cell has to produce gametes, then gametes are haploid. So diploid cell has to give rise to gametes which are haploid, then the cell division must be meiosis. So I hope you can just recall all that and you can connect it here. Okay, so diploid cell is undergoing meiosis to produce megaspores, and you should know that megaspore is developing into female gametophyte which is containing egg cell. Okay, so now you know why meiosis has to take place, because gametes have to be produced and gametes are haploid and megaspore mother cell is diploid, therefore meiosis take place. Okay, so now I told you megaspores are formed, and you know 4 megaspores are formed. Now usually in angiosperms, we will see that out of 4 megaspores, 3 will degenerate, okay, and 1 is developing into the female gametophyte. So that whole development we are studying with the help of this diagram. So here what we are showing is a part of nucellus with 1 megaspore mother cell. So here in the first diagram you can see one large cell is there, okay, that is megaspore mother cell. So what are the cells which are surrounding it? It is nothing but nucellus. So because this development is happening within the nucellus, okay. So within the nucellus this one cell is becoming, see I am not drawing the whole structure of ovule. Now you know, all of you know the structure of ovule. You know where is nucellus, right? So in the nucellus one cell, so we are drawing only this part, that is all these cells are nucellus, that is nucellar cells. In that one cell is becoming megaspore mother cell. So I told you during development one cell enlarges in size, it has a lot of reserved food materials, it has a prominent nucleus, and this one cell we call it as megaspore mother cell, okay. And this I told you it will undergo meiosis, okay. So that is what we have shown in the next diagram here, okay. The first meiotic division, okay, has happened and we see megaspore dyad. So that's the first meiotic division, and after that the second meiotic division happens and you see the 4 megaspores which are produced, okay. So now you understand it clearly with the help of diagram here. 4 megaspores are formed, and since it is a set of like 4 cells, we call it as megaspore tetrad. Now this megaspore tetrad, I told you all the 4 megaspore is not developing into female gametophyte. So usually generally in angiosperms we will see that 3 megaspores will degenerate, okay. So they are going to die, and 1 megaspore is developing into the embryo sac, okay. So this one megaspore developing into embryo sac, this mode of development we call it as monosporic development, okay. So what is monosporic development? It means that megaspore undergoing meiosis and produce 4 megaspore mother cell, okay, undergoes meiosis and forms 4 megaspores. Out of 4, 3 will degenerate and 1 is developing into the embryo sac or the female gametophyte, and this mode of development, so only when 1 megaspore develops into the female gametophyte, we call it as monosporic development, okay. So here in this diagram we are trying to focus on, that is, this is a part of ovule which is showing a large megaspore mother cell, okay, and the megaspore dyad and the megaspore tetrad, okay. So this is a series of diagram you should remember when a question is asked about the Megasporogenesis. So we explained about how megaspores are developed, okay, and I told you megaspore is developing into female gametophyte. So after the production of a single, that is the megaspore, how it develops into female gametophyte, that is what we are going to study next, okay. So a single megaspore undergoes a lot of changes, divisions, and it develops into the embryo sac or the female gametophyte, okay. So I told you 1 megaspore is developing. So a single megaspore, first what we say is it undergoes a sequential mitotic division, okay. Now you know that megaspore is already haploid because it is a result of meiosis. Now this haploid megaspore will undergo mitotic division, okay. So here in this diagram you can see the first division has happened. So megaspore has undergone first mitotic division, and you can see the 2 nucleus which has moved towards the opposite poles. After that you will see again mitosis happens, okay, and again third time. So here as a result of this, okay, you will see that a stage is, you can see a stage where 4 nuclei in opposite poles. Now here you should remember one thing, here even though it is mitosis, okay, it is free nuclear division. Means what? Now usually if you just recall, mitosis, it has 4 stages: prophase, metaphase, anaphase and telophase. So once it comes to an end, when mitosis comes to an end, you know that it becomes 2 cell, it divides into 2 cell. So first nucleus divides, that is nuclear division happens, and then cell wall formation happens and it becomes 2 cells. So here what we are seeing is only nucleus will divide, cell wall formation is not happening. Okay, so this type of division we call it as free nuclear division. Cell wall formation will happen but later. Okay, so see, after the first division there is no cell wall formation here, only nucleus divides. Again one more mitotic division takes place, again there is no cell wall formation, again the same thing here also. So after the sequential mitotic division, in this stage, that is, there are 8 nuclei. So after this you will see the cell wall formation will begin. So as the cell wall formation begins, you will see that 1 nucleus from each pole, because you see that it is a set in each pole. Okay, so 4 nuclei in each pole. You will see 1 nuclei from both the poles will move towards the center. So when it moves towards the center, 3 nuclei here and 3 here. Okay, so now you will see that cell wall formation will begin. So how many nuclei do you find there? It is 8 nucleate structure. Okay, so you can see how mitosis has happened. Okay, so here the first mitosis, then second and third mitosis has happened, and 8 nuclei are formed. Now out of 8, as I told, 1 from each pole is moving towards the center. So in each pole there are only 3 nuclei remaining, 3 here and 3 here. Now it gets organized into the female gametophyte. So how does that happen? I told you cell wall formation will begin. So here 3 nuclei you can see where the cell wall formation has started here. Here also it happens. So this is just a developmental stage what we have shown here, that is 2, 4 and 8 nucleate stage of embryo sac. Okay, after the complete development, how does a female gametophyte looks like? Okay, so this is a diagram of a mature female gametophyte or embryo sac. Now here you can see the organization of the cell. So this is a chalazal end and this is a micropylar end. So when I explained about the ovule, I told you about 2 poles. Okay, so the pole which is nearer to micropyle, we call it as micropylar end, and the opposite end we call it as chalazal end. Okay, now see here 3 cells in the chalazal end, we call it as antipodal cells. So 3 cells organized towards the chalazal end, and this is a 3 antipodal cells. The center, it's a large cell which we call as central cell. Okay, now in the central cell we will see 2 nuclei which we call as polar nuclei. This central cell has 2 nuclei which we call as polar nuclei. Coming to the micropylar end, you will see another set of 3 cells where 2 cells are called synergids and 1 cell is the egg cell. Okay, now these synergids, it has some cellular thickening like finger-like projections which you find within these synergids, and it is called as filiform apparatus. Now what is the main function of this filiform apparatus? During fertilization, it is helping the pollen tube to enter into the female gametophyte. So it is guiding the pollen tube to enter into the female gametophyte. So that is the main function of filiform apparatus. So that can also be asked as a 1-mark question, the importance or the function of filiform apparatus. So the function is to guide the pollen tube to enter into the female gametophyte during fertilization. Okay, so that is 2 synergids and 1 egg cell. Now these 3 cells together, that is 1 egg cell and 2 synergids together, we call it as egg apparatus. Okay, so egg apparatus means it is a set of 3 cells. So this set of cells we call it as egg apparatus, where it consists of 1 egg cell and 2 synergids. Okay, so the central cell will have 2 nuclei. Now this is the structure of a mature embryo sac. Now again from this part you can get a 5 marks question. One question what you can expect is a developmental stages. Okay, so the question may be to explain about the developmental stages of female gametophyte. So when developmental stages is asked, you need to draw the whole series of diagram and explain about the mitotic divisions and then the organization of the cells here. Or you may get a question only to explain about the structure of mature embryo sac. So if that is a question, you need not draw all these diagrams. Okay, only the last diagram here, this you will draw and explain its parts. Okay, so there we are ending up with gynoecium. Okay, so gynoecium, you studied about the structure of pistil. Okay, so inside the ovary there is a structure called as a carpel, or that is ovule, which inside the presence of this female gametophyte. So all these structures, okay, the diagram is important, that is the structure of female gametophyte structure and the developmental stages, and also the anatropous ovule. Okay, so now after the end of this session, I want all of you to practice structure of anatropous ovule and this series of diagram with the labelings. Okay, so practice the diagram, draw at least once. Okay, take a pic of that, and the link which is given below this video, you can open that and post your pic. Okay, so with that we come to the end of gynoecium. So in the next class I'll be teaching you about the pollination. Okay, so after studying the whole structure of male and female, so next we'll be moving on with pollination, which I'll explain in the next session. Okay, so we'll stop it here. Thank you and have a good day.